Hi, my name is Hopi Hoekstra, and I'm a professor at Harvard University. And in the first segment of my talk, I introduced you to the field of evolutionary genetics, and in particular, the study of the genetics of adaptation. In the second segment, I told you a story about understanding the genetic basis of a morphological trait. And in this third segment, what I'd like to do is switch focus and look at the genetics of a behavior. So we'll be looking at this in the context of making links between environment, behavior, and genotype. And in particular, I'll start by telling you about one behavior and how that may have evolved via natural selection. And then second, and for the most of the talk, what I'll focus on is how we're trying to understand the genetic basis of this behavioral variation. Now, one of the big questions in biology is what is the role of genes in producing behavior, and in particular, in recent years, what are those genes that contribute to behavioral variation? I think we have just a few very elegant examples of the connection between genes and behavior, which I'll highlight in this next slide. So, for example, we know that amino acid changes in the period locus affects courtship song, which in turn affects mating behavior in two different species of Drosophila. We also know that changes in the expression of the gene foraging in these adorable little Drosophila larvae affects feeding behavior. There's also a few examples of behavior genes that affect social behavior. Two of my favorites are amino acid changes in the GP6 locus seems to be associated with the number of queens in a colony. And then most recently, gene expression changes in the vasopressin gene seems to affect the affiliative behavior of males in monogamous versus promiscuous voles. Now, as I mentioned, making the connection between genes and behavior is a fundamental challenge in biology, yet I've also told you that while we have some examples, we don't have a lot of examples. And the question is, why is it so hard to find genes that affect behavior? And I just want to give you a few reasons why this may be. So there are a number of challenges to studying behavior that go even beyond the challenges that I illustrated before in studying morphological traits. For example, we tend to think that behaviors in general have relatively low heritability. That means that genetic contribution to behavioral differences may be quite low. And this may be in part because the environment can have large effects on our behavior. We know that just thinking about human behavior, for example. We also tend to think of behavioral variation as having a complex genetic architecture. And what I mean by that is that it could be that many genes seem to affect behavior. And so we're not just going after a single gene that affects behavior, but we have many targets to find. Then there's also some practical issues, such as tractability, especially if we're interested in behavioral evolution in natural populations. We have to study it in a species in which not only do we know there's natural variation in behavior, but where we can get large enough sample sizes, where we can breed them in controlled laboratory conditions with short generation times, but also that has genomic resources. But I would argue maybe the biggest challenge to studying behavior is simply the challenge of measuring behavior. So I want to illustrate that by talking about Drosophila courtship behavior. So let's say, for example, we have two species of Drosophila that have different courtship rituals. Now, I'm not a Drosophila biologist, but let me just say the courtship is actually, while at some level very simple, has a number of different components that are illustrated down below. So for example, Drosophila courtship involves two flies meeting, approaching each other, wing flapping, there's tapping, there's licking, there's smelling, there's mounting. All of this happens before copulation. So if there are differences in this sequence between species, how do we measure that? For example, do we measure the number of tapping events? And is that really the most biologically relevant component of the differences in courtship? So next what I'd like to do is tell you about how we're trying to circumvent this problem of measuring behavior. And that is not by measuring the behavior itself, but by measuring the product of behavior. And this harkens back to what Richard Dawkins referred to as the extended phenotype in a book that he published in 1982. And he defined, well, we tend to think of phenotypes as traits sort of within our own skin or within the skin of an organism. In fact, we can think of phenotypes extended outwards. For example, animal artifacts. that can be treated like any other phenotypic product whose variation is influenced by a gene. So for example, if a behavior results in the production of an artifact and that behavior is controlled by genes, we can treat that artifact just like we would any other morphological trait. So let me illustrate this by giving you two examples of extended phenotypes. So the first example that I'm showing you here is the Australian bowerbird, in which the males build these very elegant bowers. And the bowers tend to be very similar within a species. So this particular species builds a bower by putting reeds together that make this sort of U-shaped. And then he decorates it with these blue artifacts that he finds in the area. And then he'll walk back and forth, and this ritual will attract a female. But the shape of the bower differs dramatically between species. And what this serves to illustrate is that this bower is produced by a particular species-specific behavior, and the output of it is this bower that we can measure just like we would measure, let's say, the length of a limb or another morphological trait. Another example is the nest that swallows build. Again, they're very stereotyped in the type of material that's used, where they're built, in the shape of the nest. That tends to be very similar within a species but differs between species. And again, we could treat this like any other morphological trait and try to understand the genes that produce the behavior that in turn produce the artifact. So today I want to tell you about one particular extended phenotype, and that is burrows. Now, burrows have evolved in a number of different lineages. Here I'm just showing you a few examples. They've evolved in bivalves, in ant colonies, in prairie dogs, for example. And the fact that it's evolved independently multiple times suggests that it has a function. So what are the function of burrows? Well, burrows, of course, can be used for a number of different reasons, and I just want to provide you with a few examples. And these reasons can affect fitness, or the ability of an organism to survive and reproduce. So burrows can be important for predator avoidance, be important for thermoregulation, or if those species, let's say, meet and mate underground, they can be important for social interactions and also food storage or growth. So we know that burrows can have fitness effects, but the other reason we study burrows is because we have some hints that genes may be important in the production of burrows, and importantly in the differences between the size and shape of burrows. And this goes back to work done by Carol Lynch, who was at the University of Colorado, who was studying burrow building in laboratory mice. So what you're looking at here is an outline of a burrow that was excavated, and here's the entrance tunnel of a particular species of mouse. And what she showed through a series of crosses is that different components of the burrow tended to be very heritable. That is, there's seemed to be a genetic component. So we know that there's ecological relevance. We know that, at least in laboratory mice, there seems to be a genetic component, which suggests this may be a good extended phenotype, or a way to try to connect genes and behavior. The other little anecdote I want to tell you about burrows, it's one of the few behaviors that actually fossilizes. So what I'm showing you here is actually a picture of a fossil burrow that's produced by a now extinct beaver that built these burrows in this very stereotypical corkscrew shape. Well, we're not studying burrows in bivalves or ants or prairie dogs, but instead studying burrows of this particular species here, Peromyscus polionotus, which tend to live in these very open habitats, like this slide illustrates. And in fact, this is taken from one of our less glamorous field sites. It's a burnt peanut field in Alabama. But the reason we're studying this mouse is because of work that was done by Francis Sumner, who is a natural historian who early in the 1900s documented variation within and among populations of Peromyscus polionotus throughout their range. And one of the traits he documented was their burrowing behavior. And in particular, he noticed they built a very stereotyped burrow. So in a paper that was published in 1929 along with his field assistant, what he showed was that these mice, throughout their range, produced a very complex burrow that looked like this. So these burrows are characterized by an entrance hole and soil. When it's active, there's fresh soil that's excavated out of it. Then they have this long entrance tunnel, which leads to a nest chamber, and then the secondary tunnel that radiates up near the surface but doesn't penetrate the surface. So next, what I'd like to do is just tell you about how we catch these mice in the wild, which will serve to illustrate two important aspects of this burrow shape. So how do we catch these mice in the wild? Well, the first thing we do is we go out to habitat that looks like this, and we look for burrows. So here's a burrow entrance, and this freshly excavated plume of soil suggests it's an active burrow. The next thing we do is one of us, in this case my graduate student Jesse Weber, you can just see his back right here, lies down on his belly and takes some plastic tubing and weaves it slowly down that entrance tunnel. When he hits the nest chamber, he'll either blow into the tube or rattle it around. And the rest of us are not just standing around and watching Jesse, but we're at the ready, because what's going to happen is a mouse is going to pop out of that secondary tunnel, which we pounce on it, and then we've caught the mouse. Now, I've told you that the first thing I want you to note is that secondary tunnel, remember I told you, doesn't penetrate the surface. Yet we're still able to predict with pretty high accuracy where that escape tunnel is going to come out, because the shape of the burrow is so stereotyped. Okay? The second important thing to note is that secondary tunnel is actually used as an escape hatch, so that when something comes down the entrance tunnel, the mice are still able to escape. The other thing I should mention is that in the field, when we find a burrow in which the escape tunnel is visible, and we see the hole, we've never found mice in such a burrow, would suggest that once they've used that escape hatch, they then abandon the burrow. So why would these mice build such a complex burrow? Well, the answer may seem obvious. We haven't yet done experiments to demonstrate the fitness effects of different burrow designs, but suffice it to say that it's pretty clear that having an escape tunnel, especially when a major predator is a snake, makes a lot of sense. So if a snake comes in the entrance tunnel, these mice can escape out the back door. Okay, so clearly the complexity of this burrow can be important for fitness. The next thing we wanted to do is try to understand the genetic basis. So the first thing we wanted to do was try to study these mice in a controlled laboratory environment where we could minimize environmental effects and focus on the genetic influences. So to do this, what we were able to do is to bring mice from the field into the lab. Now, this work that I'm going to describe was really inspired by work by Wally Dawson, who back in the 70s, published in the 80s, showed that you could bring these mice from the field into the wild, and they would, from the field into the lab, and they would still recapitulate their burrowing behavior. So we wanted to test if this is still true. So to do this, what we built were what we refer to as phenodomes. These are, in some sense, glorified sandboxes that are quite large. So they're 5 feet by 4 feet by 3 feet high, and each phenodome is filled with about 1.5 tons of dirt. To start the trial, we put in a single mouse, and we let it live in that phenodome for about 2 days, completely undisturbed. So it's allowed to build its burrow over 2 nighttime activity periods. So just to give you a sense of what this actually looks like, here are our 10 phenodomes in our controlled laboratory facility. Then once the mouse has gone through its 2-day time period, we trap the mouse out of the cage, and to measure the burrow, what we use is what I'll refer to as phenofoam. Essentially, this is insulating foam that we can squirt into the burrow, let it harden, and then produce a cast of the burrow like the one shown here, and then treat that cast as a morphological trait, an extended phenotype of a behavior, thereby circumventing the problems associated with measuring behavior. So let me just show you in this next video of exactly how we do this. So here's Jesse Weber. He's threading that tubing down the entrance tunnel. And what you see out pops the mouse. And you'll notice that you had no idea or no indication before where that escape tunnel was until the mouse popped out. Then after the mouse is taken out of the cage, we fill this burrow with this insulating foam or phenofoam, which you'll see here. It's squirting out that escape tunnel. We thread it through, filling it with insulating foam along the way. Then what we do is we wait a few hours. The foam will be allowed to harden, and then we dig that out of the burrowing box. And then again, we treat this cast as a morphological representation of burrowing behavior. So today what I want to do is focus on just a few aspects of the burrow size and shape when we're talking about its underlying genetics. So the burrow shape, all of which we've measured off of these casts, consists of a number of different aspects. So today I'm going to talk about the total length of the burrow, shown here, and in particular the entrance tunnel length, and then refer to this as the escape tunnel, or the presence or absence of that escape tunnel. So when we started this experiment, the first thing we wanted to make sure of that these mice now 30 years or 60 generations after Wally Dawson's original study, still built these complex burrows in the lab. So what we did is we simply put a mouse in the dirt and asked, will it burrow? And as this video illustrates, these mice hit the dirt and immediately start digging. What this means to us is that this behavior is not strictly learned. In other words, these mice have never seen dirt, their parents have never seen dirt, and upwards of, let's say, 60 generations, they've never seen dirt. So the building of this complex behavior is not one that's strictly learned. So what this means to us is that there's an innate component, and in fact, there may be a strong genetic component to this burrow building behavior. Okay, there are several other very basic questions we had that we tested in the lab. The first one was, what happens if you take a mouse and put it in for one trial, and then put it in for another trial or another trial? How consistent is the burrow size and shape? Well, to do this, we simply ran that experiment 3 times in a row, giving them 2-day trials. And what we saw was that they always built a complex burrow, and that in general the length was quite consistent over trials. So what you can see here is that there is a slight trend over multiple trials, that the burrows get longer, but they're not statistically different. And this trend could be associated with either their learning to get better over time, or the first couple of trials they're still acclimating to this new environment. In either case, what I'm going to tell you, the results I'm going to tell you about are all focused on the best burrow, which tended to be the 3rd trial. Okay? Despite the fact that there are some differences, it was still remarkable how consistent in both size and length the burrows were within individuals across trials. The next question we had is, what about differences between the 2 sexes? Do the males or the females dig different sized burrows? Well, it turns out when we did these trials, we found absolutely no difference between the burrows that were built by males and females. So again, all the data I'm going to show you is going to include both males and females together. So these are some of the basics that we learned by studying these mice in the lab. Next, what I want to tell you about is what we learned about studying these mice in the field. So we sent Jesse out to the field, and he would go out into, again, this is a burnt peanut field in Alabama. He'd find burrows like this one with a fresh plume of excavated soil, suggesting it's an active burrow. He trapped out the mice and then made a cast of the burrows in the wild. So here's, after a lot of excavation, you can see a rather large burrow. For those of you who are more molecularly oriented, right here you'll see a 15 mil conical, which gives you some sense of scale. So what he did was excavated burrows like this throughout the range of these mice, and in different habitat types or in different soil types. Some that had low silt content were more sandy soil compared to those that had high silt or were more hard packed, for example, more clay. And by doing this and digging out different burrows, we learned a number of things about variation in the wild. The first thing we learned was that burrow shape is conserved. That is, in all of these different habitat types, we always have burrows with an entrance tunnel and an escape tunnel. So that shape is highly conserved. But we did see some differences. That is, even though the length of the burrows and the shape of the burrows were conserved, the depth differed. That is, when the soil got harder packed, the mice stopped digging down. But interestingly, the total length didn't change. So what that means is the angle of the burrow would change depending on the soil type, but the length stayed constant. In other words, we think the mice somehow, and we don't know how yet, are measuring length of the burrows, and that's consistent. The other things we learned, and you may have noticed this, that wild burrows are longer or bigger than those built in the lab. And there's a number of reasons this could be. For example, we just maybe didn't give them enough space in the lab, or we only gave them 2 days in the lab. But I didn't tell you one important fact about these mice, and that is Peromyscus polionotus is one of the few monogamous species. And when we go out in the wild and we catch these mice and we put tubing down the burrows, usually it's not just one mouse that pops out, but usually 2 mice, and usually a male and a female. This isn't surprising if these species are monogamous, but it does raise the question of, in the wild, maybe it isn't a single mouse that builds a burrow, like our tests in the lab, but instead 2 mice, maybe a male and a female. So I just want to illustrate what we learned about studying this in the in the lab. When we put 2 mice in a box, we ask the question, do they build 2 independent burrows, or do they cooperate and build a single burrow? And this little clip illustrates the main result, and that is the two mice build only a single burrow, and that burrow is about twice as long than what each individual burrower built. So that there's some aspect of cooperation, a term I'm using loosely here, in terms of building burrows. Okay, so let me just summarize what I've told you so far. So what I've shown you so far is that the burrowing behavior appears to have a genetic component, and this is because we've tested them in a common environment, and these are mice that, again, have never seen dirt before, so this is not a learned behavior. I showed you hints that burrowing is similar across trials, so the same individual produces similar burrows. There's no difference between the sexes, and among individuals within the species, there are actually very little differences. What we learned from studies in the wild was that burrow shape and length tend to be very consistent, but the depth would vary depending on the type of soil used. And then finally, I showed you just some hints that, in fact, in the wild, males and females may cooperate to build these burrows. Okay, but the next question is, so everything I've told you about really focuses on this one species, Peromyscus polionotus. But what we really wanted to know is how this behavior evolved. So what happens when we look at other species of Peromyscus? So what we did was we did the same types of trials across a number of different species of Peromyscus, which I'm illustrating here. Peromyscus polionotus, which I've been talking about, is shown in the bottom. So I'm just going to highlight the types of burrows that each of these species built with these cartoons. So some species built no burrows, others very small burrows, but only Peromyscus polionotus built these large burrows with this escape tunnel. And let me point out these are not to scale, so this is just to serve to illustrate an example of the type of burrow these species built. So this is interesting for a number of reasons. First, it suggests that the Peromyscus polionotus burrow, and in particular the escape tunnel, is really unique to this species. So we're looking at the evolution of a gain in complexity compared to other species in this genus. The second thing to note is that here this is showing you the relationships among these species, and you can see that even among closely related species you can have very big differences in burrowing behavior. And in particular, we were focusing on these two species down here. So even though Polionotus is closely related to Maniculatus, as indicated by this phylogeny, you can see they build very different burrows. But what's particularly exciting to us is that these two closely related species, while good species in the wild, if we bring them into the lab and give them no choice, they'll interbreed and produce offspring. This allows us to now take a genetic approach in the lab to try to dissect and identify the genes that contribute to the differences between these two species. So next what I want to do is tell you a little bit more about the burrows that Maniculatus build compared to Polionotus. So I've told you already, Polionotus build long burrows with an entrance tunnel and an escape tunnel, and this is just showing you some data on their length. By contrast, Maniculatus build these little burrows. They're like the size of a little baby sock. They're not very long. They have an entrance tunnel, again, which is shorter than the entrance tunnel you see in Polionotus, and they never built an escape tunnel. Okay? So these are very clearly distinct type of burrows. But as I mentioned, these two species, we can make hybrids in the lab. So the first question is, well, what type of burrows do the hybrids make? Well, quite surprisingly, I think, hybrids make burrows that are absolutely indistinguishable from the complex burrows of Polionotus. That is, they're long, they have a long entrance tunnel, and they always have an escape tunnel. So what this suggests is that the gene or genes that control the differences between the simple burrows and the more complex burrows are dominant. Okay. Now, to get at the genes, what we can do is take those F1 hybrids, now cross them back to the simple burrowers, and look at the next generation. So the first thing I want to do is show you some results from this next generation of mice and their burrowing size and shape. So first, let's just focus on one trait, total burrow length. So here, this is the parental generation, the F0 generation, and on this lower axis what we have is length in centimeters, and then frequency on the vertical axis. So what you can see is Maniculatus build very small burrows compared to Polionotus, and their distributions are non-overlapping. Now, as I mentioned, the hybrids, the first generation hybrids, build Polionotus-like burrows in length, although there's a few stragglers over here. So then we took these guys and bred them to these guys and asked that next generation, What types of burrows do they have? Now remember, they're going to have more maniculatous DNA in them, because these are half maniculatous, half polionotus, and we cross them back to maniculatous. And what we see is that there's a range of, or there's a distribution of lengths of the burrows. So they tend to be more maniculatous-like. Not surprising, they have more maniculatous DNA, but the important thing is we still get mice down here that are building polionotus-like burrows. So this distribution tells us two things. One, it's not a single gene. We don't see a 3-to-1 segregation of long and short burrows, that there are multiple genes. But because we can recapture the polionotus-like burrow, this suggests that there's a handful of genes and not hundreds of genes controlling it, which is quite exciting. So the first result is there are multiple genes, not hundreds, maybe a handful, that contribute to differences in burrow length between the two species. Now, this is just length. What about shape? So now we're focusing on that second generation again and asking what shape burrows do they make? Well, they make a range of shapes. So some build these very small burrows like the maniculatous parent. Some build burrows like this one shown here, which is like the polionotus parent. But what you can see is sometimes we have burrows that are small with an escape tunnel and other ones long without an escape tunnel. And if we simply count up the number of individuals that build an escape tunnel compared to those without an escape tunnel and asked how does this segregate in that second generation, what we see is there are almost equal numbers, okay? This is not distinguishable from a 1-to-1 ratio. What this suggests is, or is at least consistent with the role of just a single gene controlling these differences, in controlling the presence or absence of the escape tunnel. Now let me say this is data early on, and we've done more experiments that suggest that maybe it's a little bit more complicated, but again this suggests that maybe the presence or absence of the escape tunnel has a relatively simple genetic basis. Okay, so what I've shown you is that in terms of size and shape of the burrow, we're looking maybe for a handful of genes. But of course the big question is what are these genes? Now to do this, we took all of these mice which we've characterized their burrow building behavior, and now we've genotyped them at a number of markers throughout the genome, and asked is there a correlation between what regions of the genome they get from the complex parent and the simple parent in terms of burrowing, and the burrowing that hybrid exhibits. So here what you're looking at is a linkage map. I showed you one of these in segment 2 as well. So each one of these lines represents a different chromosome. Each one of the dashes represents a different marker that tells us whether it comes from the maniculatous parent or the polionotus parent. And the main thing I want you to take away is it seems like there are 4 regions of the genome indicated by these black arrows that seem to control burrowing behavior. So again, this is consistent with what I've told you before, that there's probably a handful of genes involved. What's interesting, again, is that it seems like some of these genes, like this one shown here, seems to be largely associated with escape behavior, which these 3 other genes seem to control burrow length. Again, consistent with the results I told you before. Okay, but what are these genes? So I'm just going to focus on some very preliminary data and tell you about how we can go from this resolution down to the resolution of genes. So now what I'm going to do is I'm going to focus on this one right here. And now I'm taking this chromosome, I'm going to turn it on its side. So here's the chromosome, here's position along this horizontal axis. And on the vertical axis we have LOD score. You can think of this as the statistical association between genotype and phenotype. You can see as we move down the chromosome, we get to this peak of, that statistically significant peak of high LOD score, which suggests that this region of the chromosome has a gene that seems to influence burrowing behavior, and in particular length of the burrow. So and here are our markers shown below. Okay, so now let's narrow into this region and ask what genes occur in this region. Well, there are a number of genes, about let's say about 50 or so, but there's one in particular, the one I've highlighted in red, that we're excited about. So here are a number of genes that occur in this region. These are just the ones that are brain expressed genes, which we think are especially good candidates. But next what I want to tell you about is just a hint of a type of gene that may be involved in this behavior, and that is what we refer to as Chrome5. So why are we so excited about this particular gene, Chrome5? Well, it's based in part on what we know about this gene from studies in both mice and humans. So this muscarinic acetylcholine receptor, 5, has been studied in laboratory mice. We know it's expressed in the brain, in particular in dopamine-containing neurons in the basal ganglia. It's expressed in particular in two regions of the brain that are associated with reward circuitry and another region associated with motor control, spatial learning, and addiction. We know that in human studies, in genome-wide association studies, the same gene has been associated with nicotine addiction in humans, and if we knock it out in mice, it's associated with morphine addiction. So our current hypothesis is that these mice that build longer burrows may in fact be addicted to burrowing. Now we haven't proved this yet, but we do have some tantalizing preliminary data. So we've looked at the expression of this gene now in our Peromyscus mice and have shown it's expressed in the same regions of the brain that has been shown in laboratory mice. But particularly exciting is that there's a difference in the expression level between the polionotus and the maniculatus. In particular, it's expressed higher in the complex burrowing species. So this, while we still remains to be shown whether this gene is truly involved in these differences in burrowing behavior, this does give you some sense about how we're going after the genes that are involved, and ultimately to functionally prove that they are causally controlling behavior differences. So in the second part, I've shown you that burrowing differs among closely related species, which is quite exciting because we can take advantage of this to try to get to the underlying genetics. I showed you that the polionotus complex burrows are unique, so we're looking at a gain in complexity of this behavior. And because we can cross these closely related species, we've shown that F1 hybrids produce these complex burrows, suggesting that the gene or genes are largely dominant. And then I showed you with continued crosses in the second generation that complex burrows may be controlled by just a few loci compared to the simple burrows. And that different aspects of the burrow seem to be controlled by different genes. That is, there are different genes that control length of the burrow compared to presence or absence of the escape tunnel. And then I've hinted at least a type of gene that we think may be contributing to these differences in this natural occurring behavior, which may be one of the, we may be on to one of the first examples of a gene controlling a complex mammalian behavior. But I want to end today by telling you about the work we're doing, not just to understand the genetics of the burrow architecture, but actually what the mouse is doing. So the last question I want to ask is, how did the mouse's behavior evolve to result in these different extended phenotypes? In other words, do the complex burrowers, Peromyscus polionotus, do they dig at a faster rate? Do they dig more efficiently? Do they dig for a longer period of time? Now, answering these questions is actually quite complicated because these mice are nocturnal, they're doing their behavior underground, so how do we study this? Well, my postdoc, Brant Peterson, has come up with a nice ingenious way to study the mouse behavior more directly. And that is, he built what we refer to as an ant farm, or what we refer to as the Brant farm. The Brant farm is essentially a two-dimensional burrowing structure. So we have plexiglass on either side, it's filled with dirt, and it's about the width of a mouse. We can illuminate this burrowing structure with infrared lights, and then we can record the mouse behavior. So I just want to give you an example of what a mouse burrowing looks like. So you can see here the mouse is digging its burrow. It's gotten to the point where it's starting to dig out its nest chamber. It turns on its back. It starts digging upwards. And so by videotaping them in this new setup, we can actually watch exactly what the mouse is doing. Now we got really excited about this particular approach because it was another way to study the genetics of not just the extended phenotype but the mouse behavior directly. But one of the things that we learned very quickly, that while it's very easy to capture these videos, actually scoring these videos became very hard. So this goes back to this problem of how do we measure behavior. So it takes a lot of graduate student, undergraduate hours to actually watch these videos. But luckily, Brant, again being ingenious, came up with another idea, and that was to automate this behavior. So here what you're looking at is another mouse digging in the ant farm again, but this time it's all completely automated. So there goes the mouse down the hill. We can watch this, record this over time. We can look at the burrow shown here in red. We can see where he's about to dig shown in blue. We can also measure the change in the slope of the hill, so the amount of digging, excavating is correlated with the amount of sand that comes out, etc. So by automating this, not only is it a lot simpler, doesn't require so many man hours, but we also do this in an unbiased way. So I just want to show you some results from this output and hint at some some early results from this experiment. Okay, so what we're looking at here now is the result of that automated behavioral analysis. So here what we're looking at on the bottom is time, and time is color-coded. So you can see throughout the course of the night we get to more blue. So what you're looking at here is this slope, and here's the tunnel or burrow that the polionotus dug. And then you can see over time the soil that's excavated goes up. And this cloud-like images tells you about where the mouse is spending its time. So the denser the cloud, the more time a mouse has spent there. So you can see the mouse has been running up and down. It's also spent time digging out its burrow. But what's really neat about this is we can compare what a polionotus is doing compared to a simple maniculatus, and there's some very outstanding differences. The first thing is you can see on this axis, which is time, above a line is showing you when they're digging. Below the line is activity that's not digging. You can see the mouse is active throughout the whole night, but this mouse starts digging immediately during the night when it's put in the dirt box, and then finishes its burrow early on and maybe does a little bit of fine-tuning later in the night. But this is very different from what a maniculatus does, whereas a maniculatus that builds a teeny little burrow shown up here, what it does is that it doesn't do any digging to about 2 hours before lights come on, and then it digs its burrow very quickly, and then it almost doesn't do anything else and just sits in its burrow for the last hour before the lights come on. So this is just to illustrate that these are new features of the mouse's behavior that we're starting to uncover that we never would uncover by just looking at the extended phenotype alone. And so these are other traits that we're now trying to dissect genetically, just in the same way that we've been looking at burrow architecture. So what I hope I've done today is to convince you that this is a very exciting time in the field of biology, in the sense that we're now at a point where we can start to make these links between environment, behavior, and genotype, understanding how those behaviors evolved in the wild, and also the genes and how those genes work through neurobiology to produce variation in a behavior that's important for fitness. Before I end, I want to acknowledge the people that did the work. As I've hinted at, Jesse Weber, a former graduate student in the lab, worked on the genetic basis and also did some studies on cooperation. Wenfei Tong was a collaborator on the cooperation study. Brant Peterson has been working on the genetics and the mouse behavior, and a graduate student, Hilary Metz, has been working on the genetics and starting to work on the neurobiology of this behavior. But I would say everybody in our lab group, shown here, at one point or another has gotten their hands dirty, quite literally, by helping us dig out burrow casts. We've also been lucky to have an amazing team of undergraduates that have helped with this project, as well as various funding sources. So thanks again for your attention, and thanks for joining me today.